Okay, so this is our objective, okay, is to really begin to understand exercise. And really, some of you who are involved in this, I don't have to tell you that exercise is good for you. We all know that. One of our goals is to really understand, this is one of our patients, Bernie, and Bernie has Parkinson's disease, and let me just... And he has all the typical features that we see in a patient with Parkinson's disease in terms of the gait. I don't have to tell you that this is a patient with Parkinson's disease. We can all acknowledge his slowness, the akinesia, the bradykinesia, the freezing that occurs, all those typical features that we see in patients. This is not a different patient. This is Bernie. This is Bernie after 6 or 8 weeks of intensive physical therapy, focusing on gait, focusing on balance, and focusing on a number of features. That's not the same person. That's a very different course of disease. I am not preaching that this is a cure. It is not a cure. If someone tells you a cure, tell them there's a place in Washington that you can go to take over. But it's definitely not the cure. Oops. You know, the equipment, you're balanced. We all know exercise is good for you. And on the way up here, I was thinking, I don't have to preach that. We all know exercise is good for you. And I'm trying to think, the only time that I can think of exercise being bad for you is jogging in a minefield. That's about the only time that you're going to see that as a problem. We really want to understand the fact, specifically, what are the mechanisms? What's happening within your brain? And can, in fact, we promote the benefits of exercise in some particular fashion? And then especially in the context of Parkinson's disease, how can we use exercise to target beneficial brain changes in disease to either modify disease or improve your quality of life, but eventually to discover and better understand the underlying mechanisms so we can hopefully find a cure someday. That's really our goal. So I'm not an exercise physiologist. I don't study exercise in the sense of seeing if it's good for you or not. We know all that. I want to know why, and I want to know what the underlying molecular mechanisms are so that we can improve it. And especially when, you know, those of you who want to take a nap, go ahead right now. There's a slide that I have that's kind of like highlights everything. Is to really tell you what type of exercise you should be doing and why you should be doing that type of exercise. And so that's what I'd like to leave you with in this particular discussion today. And really, we kind of we've been studying exercise since like 2002. When we first started looking at this in the laboratory. And we've kind of began to look at expanding from a couple of regions that nobody else does. And so we bring something rather new to the table. And the two things that are really important for exercise that we've been studying in our laboratory is understanding cognition and metabolism. Why is thinking important to you as a patient, to you as someone who's trying to get the benefits of physical therapy from exercise? Why is the thinking part so valuable? This is really something that Dr. Petzinger has been pushing for the last decade or so, and we're really getting some amazing understanding. The other thing is understanding what changes are taking place in the brain. This is what Adam will talk about, metabolism. How are you making energy, and how can we tap into that? Who is making the energy, and what are the circuits that are involved? Can we make that better? And that's really something that his work is really showing. And the whole idea here is, can we in fact get restoration of normalcy? Can we get motor behavior? Can we possibly modify disease progression? We all know we probably are modifying disease progression. I've been running the LA Marathon with some friends. There's one guy, John Ball, you might know. He's had Parkinson's since he was 37. Last marathon we ran together, he beat me by 2 hours. And he's 72 now. So he has modified disease progression in some particular fashion. I don't have to tell you what Parkinson's disease is. We know that it's a loss of the dopaminergic neurons that reside in the substantia nigra parasimpactum. You have a failure of delivery of the neurotransmitter dopamine to the basal ganglia. We all know that. Okay? You all have had those pamphlets. What we do know, in fact, is that there is a cognitive aspect to Parkinson's disease that we have been thinking about for the last 10 years or so, and beginning to now get emerging data that's really going to be beneficial. We do know, for example, that in the context of Parkinson's disease, there's a loss of automaticity. And I know many of you who have Parkinson's disease, you really have to think about everything you do. It's sort of like, I got a sore hip right now, but I have to think about every step I go up. I have to think about going on the sidewalk. I have to think about getting in the car because I've got this confrontation, I've got this challenge in terms of something that's sore. You as a Parkinson's patient have to think about everything. Those factors that used to be automatic are no longer automatic for you. What we'd like to do is to see if we can get restoration of that automaticity. Can we put back something that was automatic and get to some restoration? And in fact, we know that freezing is a cognitive challenge. It's not that your neurons aren't working fast enough, because that doesn't make any sense to us as a physiologist and biochemist. It's the processing of the information. It's the challenge that you have. It's like anytime somebody walks into a new museum, you walk into the Broad Museum in downtown LA and you stop there and you look at everything, all right? And you're just like, oh my God, look at all this. Now, a neurologist will look at you and go, that person just froze. Right? They're standing there frozen. No, not frozen. I'm processing this information so that I can now move. And this is what you do in the context of Parkinson's disease. You are processing this information of going from linoleum to carpet, to go from the kitchen to the living room, to go from the car to the doctor's office. You're processing information. And what we'd like to do is to see if we can find a way to actually restore that processing of information, to actually begin to target parts of the brain that we don't traditionally think are as important for Parkinson's because we are told it's a motor disease, right? It's just a motor disease. No, it's a thinking disease. It's actually a big part of our thinking. And why do we think that? We think of that because we're human. And one thing that's absolutely unique about the human body is the fact that some of you remember, that actually about 7 million years ago, we transitioned from walking around on 4 feet to walking on 2 feet. And yes, there are some Saturday nights that we end up back here, but that's a different issue. I'm not getting that right now. That is a gigantic computational challenge. Huge amount of information is needed to move on 2 feet. It's easy to make a robot that walks on 4 feet. It's easy for a baby to transition across the carpet on 4 legs, or 2 arms and 2 legs. That's easy. But to stand up and now begin to balance is a huge challenge. You have about 600 muscles in your body. How many muscles do you think you have to control when you walk? Exactly, 600. You've got to turn ones off, you've got to turn ones on. 600. That is a gigantic computational load. That is huge. That requires some very much specialization. God forbid you start doing something talented with that. Okay? Whether it's skateboarding or gymnastics or soccer or going upstairs or going on the Stairmaster. Oh my God, that is a gigantic cognitive load. So, what have we done? We've created what's called the prefrontal cortex. So we have a part of our thinking, and actually a third of your brain is dedicated just to the thinking of movement. And that's really all your brain really does. Just to make things really reductionist for us. The human brain is developed for only one purpose, and that purpose is to move through space, to transition from one spot to another successfully, to go get food, to go get a mate, to run away from a saber-tooth tiger. That's all the brain does. Luckily, it's got a bunch of other fun things that just came along with it. Language, music, empathy, art. Those are all what we call strangles. Those are just kind of like the advantages of the human brain. It's kind of like, nice thing. It's like discovering something really good, like a stereo that you just bought. Oh my God, look, it's got an on-off button and a treble. Now and the bass. Oh my God, this is amazing. That's exactly what the human brain does for us. We've known for decades exercise is good for you. Okay? We can then begin to ask, what is it that exercise is beneficial for us in the context of using animal models? And so in the laboratory, we can give rats and mice Parkinson's disease, basically, by using a very specific toxin or surgical technique, and they can destroy the same cells that are lost in Parkinson's disease. And so we can then begin to ask, if I were to take this animal, and there's one animal up here, and there's another animal up here, they're both Parkinsonian animals, and we run them on treadmills, and after about 3 or 4 weeks, they kind of look normal. They actually look identical to each other. But yet one doesn't, one's Parkinsonian and it's been exercised, and one's Parkinsonian and been exercised, and the other one that isn't exercised and stays back here. So we can actually show the benefits of exercise. And we think, those of us who are in physical therapy, how many have had a knee replacement? Okay? So you have a knee replacement, you're too young to have a knee replacement. Okay. Same one twice, or do it for each one? Okay, make sure. Good deal. The idea is that when you have hip replacement or knee replacement, we put you back into physical therapy and we ask, let's get the system to go back to where it was before. Let's get it working just like it was before. So in 2002, we had a postdoc who came to the lab, and one thing that she was interested in says, oh, Mike, you have these mice, you make them Parkinsonian. I study treadmill running with humans. What if we took your mice and ran them on the treadmill? And would we get them to recover faster? Could this become our physical therapy that we can actually enhance recovery in our animal models and mice to make them that way? And yes, we did. And so when we ran them on the treadmill, they actually get better. And they get restoration of all those things that we saw that was lost that they had with the Parkinsonian. And then we expected, in fact, that the brain, in response to the new training, would just be forced to go back the way it was. Just like physical therapy forces you on the treadmill or on walking to then go back to what it was before so that you can walk the same way. We see the restoration of a number of neurons, and this is just a type of stain that we use in the lab to go in and look at the brain. Is it the same that we saw before? In fact, it showed a lot of features that were identical to it. But when we start looking at some of the features of the basal ganglia, that part of the brain that's compromised and damaged in Parkinson's disease, rather than seeing it restored to what it was before it had Parkinson's disease, we did not see certain markers going back. It's like one of your radiologists and showing a picture of your brain, and he looks at your picture of your brain and he goes, you know, all this wonderful dark stuff here, which is representing dopamine, isn't there, and it's not that brain. So that must be a patient, that must be somebody who has Parkinson's disease, because that's the standard that we would use. And when we looked at a number of other features, like the amount of dopamine, because we've been told as neurologists for the last 100 years, exercise puts back dopamine. It doesn't. Okay, that's based on a bunch of dogs running on a treadmill. It's not the same thing. You can't extrapolate that to humans. And in fact, what we found, to make a long story short, is that in fact, what little dopamine was left was being used in a different way. And we have some very sophisticated techniques that allow us to do that. So we can look at some specific neurons that are in parts of the brain that control motor. And by driving this little mouse to run, and then asking, do you have dopamine coming back? No, you don't. Oh my God. But what little dopamine you have, the 5% left, the 10%, are you using it differently? And in fact, that's exactly what happened. It was using what little dopamine had differently to then succeed in terms of function. We saw dopamine receptor changes taking place. This little red dot here represents the amount of dopamine receptors that were present. They're starting to come back. And who gives a rat's ass about a rat? Right? We want to know if this is relevant for humans. And so we can then look at humans and we can run them on a treadmill, just like we can run our rodents on a treadmill. And we then looked at the human brain, and in fact, we saw the restoration of the same things. Of course, this patient didn't give me his brain. Okay, but we can look at it through indirect mechanisms like imaging. And when we imaged the dopamine receptors, we saw restoration of the receptors just like we saw in the mouse. Which means to us, and began to tell us in 2004, that in fact, the brain has found a new way to solve a problem in relearning motor behavior. And this is what we call neuroplasticity. Brain change, the plastic of the brain. It's opened up a whole new field in terms of our understanding of what it is that the human brain can in fact do. When we first published this, the NIH study sections really just kind of told us this was all BS. It was impossible. Why would you even be interested in neuroplasticity? Because we do know that, in fact, the brain with Parkinson's is deteriorating and it's only going in one direction. Why would it be at all interested to begin to drive other mechanisms that would get restoration because they're not there? And basically, we just said this is complete bull. And so they told us it was bull. So we said, good bull is bull. And so we countered back and said, no, we believe that, in fact, neuroplasticity can serve as a very important target in getting restoration of some features within the human brain. No, it's not the cure yet, but certainly if I can give you, or if we can give you, or a physical therapist or neurologist can give you a quality of life that may be very different than you would without it, I think that's very important. We certainly have many patients who are in their third decade of Parkinson's. 30 years. Some of them are even using walkers. It's amazing. And so, in fact, is there some hope in terms of understanding them? This whole idea of neuroplasticity is not new. This is not Dr. Jackie. Dr. Jackie discovered neuroplasticity through the Nobel Prize, and we got something on it. This is 200 years old. Lots of old literature. But one thing that we found was very interesting was the fact that one person in 1929 published a book and said something that I read, I have copies of the book, that was fascinating and really told me something that was very important. Because we'd all thought the brain is like your house. You build it, and you live in it, and you deal with it, and parts fall down, that's just the way it is. Dr. Campbell said, no, that's not quite the way your house is. What is happening is that the stability that is maintained is excitable and capable of modifying itself according to external stimuli and adjusting its response to the stimulation. It's dynamic. It's modifiable. It's always has to be kept going, kept going, kept going. And in fact, if you stopped and you don't use it, you're going to lose it. But we maintain its functionality by constantly using it and constantly engaging it. And that was kind of something that we thought, this is going to be actually very important. And then what happened is that this began to bring in the whole idea of cognition, because we do know that there's this huge connection between the motor circuitry in the brain and the thinking part of the brain. They go hand in hand. And we have a lot of evidence for it, but I just want to see if I can just quickly tell you an example. And one good example is the idea that learning a new sport. We can learn tennis either at a very young age. We can learn tennis, and this is a picture of me when I was on the circuit years ago. This is how I can afford things. Or we can learn it for the first time later in life. And we do know that, in fact, when you are learning, what's the most important thing that you do when you learn something new like this? You got a coach. Okay? You got something, you keep a score. You start getting feedback, being told all of these things, constantly being told. Okay? And this is very important because it, in fact, is one of the parameters that tells us that you are cognitively engaged in that particular effort. You're thinking about what you're doing. So, for example, if you wanted to learn tennis, and you got on the court with these guys, and you're distracted, okay, and you're like, I can't focus on this, I can't, you know, I'm either looking at him or I'm looking at her, and I'm distracted. I'm not going to learn. What you're doing is you're disrupting the thinking part of your brain. And so this lack of focus ends up as a lack of learning. And this lack of learning is the mechanism that we believe is important in mediating the changes within the brain that will lead to long-term changes that are long-term permanent circuitries, long-term behaviors. And that's what we do. Think back when you're 1 year old. Think about your grandchildren, getting them to walk. All right? They're learning to walk. You're giving them feedback. It's a learning modality. Okay? Walking is something they learn. Riding a bike is something they learn. And then eventually these learned behaviors become permanent. And so we really want to understand that. This opens up and reveals to us a very important parameter. And that's the idea of something that the patients will always be asking us is, what type of exercise should I be doing? Okay? Should I be doing the Argentinian tango? Should it be the box trot? Should I be, what exactly should I be doing? We stumbled upon this whole idea of linking between cognition and the metabolism of neuroplasticity. What is it that's linking these two? And one thing that was very important was this whole idea of thinking of aerobic exercise versus skill-based exercise. Skill versus aerobic. Skill versus aerobic. Think of it. Aerobic exercise. 24-hour fitness. Okay, you go to 24-hour fitness, you see somebody on the treadmill. Okay, they're on the treadmill, really slow. Watching Fox News, okay? All right, their brains are not engaged at all, okay? And it's purely aerobic. Now think of someone who's doing something more highly skilled. Tai Chi, for example. Those of you who do Tai Chi, you come up sweating. It's not just standing there like this, okay? It's really quite intensive. So, is there a difference between the two? We certainly know that, in fact, the difference between the skill-based exercise and the aerobic exercise is really what you engage and what parts of your brain that you engage. If anybody's interested, you can get these slides off our website, so don't worry about making notes and things like that. I'm happy to send them if you can't get them down, or you can get them yourself. But the whole idea is that we have exercises that are skilled and exercise that are aerobic. And what's the link between the two? We live in a continuum, so I can't really delineate the two very effectively because it's a spectrum. The only way I can get you to actually just do aerobic exercise would be to cut your head off, so you're not thinking about what you're doing. Most IRBs don't like that, and so we tend not to do that type of intervention. But we do know that, in fact, when we look at the differences in terms of the circuits that are engaged between those types of exercise which are highly skilled versus those that are aerobic is that we get these other areas of the brain, especially those that are involved in the thinking part of the brain, become engaged. So, how can we get the mechanism? We can actually do this with rats. And in fact, with rats, we can put them on different types of exercise regimens. And these little exercise regimens and exercise patterns can be either skilled or non-skilled. And a skilled version is shown here, the non-skilled. The non-skilled is just simply a motorized running wheel that's just nice and smooth. It's like 24-hour fitness. We put Fox News on for them and they just kind of walk along. Okay? And they're not thinking because it's no big deal. It's really trivial. These rats, in fact, these rats here are in fact challenged because we've taken out this nice smooth rim, we've taken out some of these rungs, and they have to sit and think about what they're doing. They have to be cognitively engaged in their exercise. So they look up at the students and say, hey, what do I do? And they now are told to grab, hold, and they can then begin to do that. They're actually engaging parts of their brain. Okay, that's what you think's going on, right? Well, we need to show that. We can show that through a number of different techniques, including imaging in animals. There, they give us the brain, which is really nice of them. And we can look at the imaging of their brains, and we can actually see, if we compare those non-skilled aerobic versus the skilled exercise group, we actually see that their thinking parts of the brain, the prefrontal cortex, the cognitive part of the brain, that's usually not involved in motor, is actually being engaged. And these animals, in fact, show greater benefit from the exercise that is skill-based than those with just the aerobic. So we can, in fact, get validation of our approach. Okay, who gives a rat's ass about a rat, right? Okay, we need to look at this in humans. We see the same thing in humans. This is a study from Dr. Petzinger that's beginning to show that, in fact, when you have certain forms of exercise in humans, you actually engage those parts of the brain that are part of the thinking. And what's most important about her work was the fact that if you were to disrupt the thinking part of the brain, just like when you're trying to learn tennis and you're not focusing, okay, you're distracted. If you have, for example, mild cognitive impairment or having thinking problems, and this could be due to disease, this could be thyroid, this could be blood pressure, it could be a lot of different things. If you have problems and challenges in the thinking part of your brain, you will not get the benefits as you would if you were to have clarity in that thinking. So in fact, cognition is as important in terms of targeting, in terms of getting benefits from the interventions as the motor. So it's extremely important that we look at both. And so this is really fundamentally important that in the context of the Center of Excellence you would look at and see your neurologist, your movement disorder specialist, would say you need physical therapy, but I don't want you on a treadmill watching Fox News at 24-hour fitness. I want you to be engaged by a neurophysical therapist who actually has developed a number of programs that will engage those very specific circuits of the brain. And this is exactly what we see. So this raises the final, this is the last couple of slides here, and then I'll have Adam come up. This is fundamental. Anybody's napping, wake them up. Okay, this is the one slide that they should actually see. Okay? And this is the slide. The slide is really asking, doctor, what type of exercise should I be doing? Okay? They offer all these different options. They offer Tai Chi, treadmill training. This is my favorite, of course, tango dancing. Boxing, amplitude training, a whole series, walking through the park, all these different, what should I do? We do now know based on our animal studies and some of our studies in humans now, we can tell you that the type of exercise that you should be doing has to be skill-based exercise. It must be a learning modality. Challenge yourself to learn something new. It must have a high degree of intensity. It must have a high degree of specificity. Gait, balance, let's work on walking. Let's work on running. Let's work on trail walking. Let's work on treadmill. Let's work on Tai Chi. That's these types of things. That's specificity to us. We also see it must be difficult, meaning challenging. You have to get the system to learn. It's got to be challenging. Those of you who are trying to relearn Latin or play the violin, oh my God, that's challenging right now. Okay? But later on it'll become automatic. And it also must be complex. And it must be complex in terms of engaging all these other circuits. Complexity means thinking. And so therefore we're engaging the motor aspect and the thinking aspect. And together we want to build that bridge between those two systems, because that can give us restoration. So exercise must be a learning modality with goal-directed targets and cognitive engagement. The The mechanisms involving metabolic changes act to consolidate specific circuits, and this is what Adam will share with you. It has to be a learning modality. Okay? So, if you walk away, and it's not saying that boxing is better than this, that tango is better than this, that tai chi is better than this. That's not what it means. Okay? Those of you who are involved in boxing and tango and dance and tai chi, you know, sometimes directly and indirectly, that this must be a learning modality. Make it something that we can learn from, and that's when we start to see the benefits of the brain. And this is when we see the benefits. We're learning to, we're relearning and then walking. So what I'd like to do now, and we'll for the last 15 minutes of our talk, is hand this over to Adam. Adam is also is a graduate student in the laboratory, and he is involved in trying to understand the mechanisms that, in fact, begin to identify why and what is changing within the circuits of the brain. And could that, in fact, give us validation, give us evidence-based medicine of why we should do certain types of exercise, and how much we should do, and what the impacts. But also, it's helping us to identify potential therapeutic targets, because there may be situations where we, in fact, can add some drug. Something as simple as cinnamon is able to elevate you to a point where you can become engaged again. And we know that that engagement is extremely important, and we know, for example, that social groups are as important as exercise groups. Okay, and we discovered this a few years ago. You can't take people to sit around and whatever. Actually, being social is cognitively engaging, much more so than you think. Whatever you may say it is. That's why I said going down to the bar, watch the Danish guys. That is cognitively social engagement. That's exactly why we did it.
So my exercise. So when I was an undergraduate at Yale, I had the opportunity to work at the Glasgow Institute at UCSF, where I worked with Dr. Leonard Mookie, and we were studying astrocytes in the context of Alzheimer's disease. So what I learned here, working with Dr. Anna Orr, who is currently running a lab at Cornell, studying both Parkinson's and Alzheimer's disease, that these cells, these astrocytes, are able to modulate the ability for animal models of Alzheimer's to learn and actually retain memory. And we saw a lot of the same changes we saw in the animal models in humans as well. So this is the first time for me where I learned that things beyond the neurons were able to actually modulate the brain function. This got me really interested, and when I came to USC, I was able to have the opportunity to expand upon this knowledge that these cells are able to contribute to the ability for the brain to have neuroplasticity. So as Dr. Jacob mentioned, there's a loss of neuroplasticity in disease, and we are seeking to restore that. The loss of that is at synapse. And synapse, of course, is where neurons talk to each other. It's the physical interaction between two parts of the different neurons come together to send a chemical message, and that perpetuates the information processing in the brain. So we see a loss of these synapses in animal models of Parkinson's disease, worked by a previous grad student called Toy in our lab. And exercise actually is able to restore these things. So all these little dots you see here, these are all little synapses. These are all little connections to other parts of neurons. And so this was encouraging for us to show that the brain is actually physically remodeling itself to mediate these new connections and to continue to relearn motor behavior. Next slide. Sorry, okay. So when we talk about exercise-induced neuroplasticity, keep going. Yeah, so as Dr. Flack mentioned, and he's kind of giving you this whole rundown, that we do see a lot of these changes both in the behavior of these mice. See, one is with Parkinson's, with exercise, and without exercise. And we do see a number of different structural and chemical changes in terms of dopamine and the actual structural changes of neurons, but we are continuing to ask what the underlying mechanism is of these circuits and what is actually causing exercise to do all these positive things. And so, so astrocytes themselves are integral to the functioning of these synapses. So we lose synapses in disease, and we're restoring synapses with exercise. When we actually see and study astrocytes, we understand that they release a number of different molecules that actually are facilitating the formation and the maturation and the degradation of synapses. So they themselves are important for all these benefits we're seeing in the brain. As well as being important for synapses, they also do a lot of other things. So all these blue things here are all the different things that astrocytes do in the brain, which is a lot. But what I'm most interested in is energy substrates, especially lactate, and how the metabolism of these cells, both centrally and peripherally with your muscles, may contribute to neuroplasticity. So again, as Dr. Jacob mentioned, we have different types of ways to think about exercise. We have skilled exercise, so this is me actually in college running the hurdles, and this is the highly skilled exercise, versus Dr. Jackwick's son, Nicholas, running a half marathon here, which is highly aerobic, right? So these are two different ways in which we can study how astrocytes are responding to exercise and maybe facilitating changes in different parts of the brain that are responsible for these different types of exercise. So how can we link exercise and cognition and neuroplasticity and metabolism all under the umbrella of improved motor behavior? What we can do, and the best way we think to study this, is looking at what we call the neurovascular unit, which is the combination of blood vessels, so vasculature in the brain, where astrocytes, which mediate blood flow as well as mediate synaptic plasticity and neurons themselves. So the combination of these three cell types, form what we call the neurovascular unit. So we believe that there's a specific coupling that occurs between all three different of these cell types in very specific parts of the brain that are facilitating the actual improvements in motor behavior and cognition with exercise. So if we look at the neurovascular unit, this is from a recent review paper of ours, we can see when you're not exercising, there's very little activity in these circuits, and cells are just doing what they do. Neurons are doing their thing, astrocytes are doing their things, there's very little blood flow in the brain to these areas. But when we think about what is actually happening in the synapse, all these white arrows is where energy is being used. So there's a lot of things happening just to allow two neurons to talk to each other. And that metabolic burden is heavy here at the synapse. So in your brain, about 80% of all the energy that's consumed is consumed at these little intersections between two cells. And astrocytes are important for mediating a lot of these changes and a lot of these metabolism. So when you exercise, next slide. Thank you. We have increased blood flow, and this really ramps up the whole system. It gets a lot of metabolism going, and allowing these two different cell types, the neurons and these astrocytes, coupling with blood flow to actually improve metabolism and increase the actual metabolic capacity and the neuroplasticity that's occurring to meet this new increased synaptic activity demand that's occurring when you exercise. And when we look at actual gene expression changes in the brain following exercise, we see with increasing amounts of exercise, either no exercise, 1 day, or 5 days of exercise, we see increases in a number of different genes, keep going, that are related to metabolism. All of them go up over time. We see all these genes are being upregulated to increase blood flow, to increase metabolism, to increase the ability for these cells to continue to produce energy to facilitate the neurotransmission that's occurring with exercise. So what I did to study astrocytes and exercise was we took mice and we exercised them, and then we looked at different parts of the brain that are involved with cognition and exercise, and then we studied how these cells change. So these cells are very dynamic, so they change their structure a lot, and they do so in response to changes in the brain itself. And so what we can see is that when we exercise, there's very dynamic changes in different aspects of these cells, and we can see here in this reconstruction of these cells that they actually are getting bigger or smaller depending on how much exercise you do and where they belong in the brain. And this told us that these cells are actually very able to be modified by exercise, which was new for the field to understand that these cells are actually dynamically undergoing change. And these changes might be reflected in actual functional changes as well. So this metabolic cooperation I was talking about, this coupling of neurons and astrocytes with blood vessels during exercise, as we showed, this is very energetically demanding. One way in which astrocytes are able to allow this to go on, as I said, is through lactate, this energetic substrate. So we usually think of lactate when you're exercising, you work really, really hard, and your muscles produce lactate, and that's what makes you sore, right? You have lactic acid, things like that. Well, the field has recently kind of gone through a little bit of revolution in the sense that lactate itself is no longer a bad thing. It's actually incredibly important. And one way in which this may be happening is that astrocytes themselves can create lactate within the brain, and neurons can use that actually to improve their performance in their actual metabolism to allow for neurotransmission to occur. And what's been shown is that when you delete the ability for astrocytes to actually make lactate, or the brain to actually use lactate itself, you get a lot of different problems. You have problems with memory consolidation, you lose the ability to have exhaustive exercise capacity, you actually lose long-term memory information as well. And so this cooperation, especially with lactate between these two cell types, seems to be increasingly important. And lactate itself is highly relevant to exercise, because when you're exercising intensely, and we know that intensity is important, as Dr. Jackwick mentioned, lactate is being produced, and it goes up into your brain, and it doesn't come out. So something's going on in the brain with lactate that is being used to facilitate a lot of these changes. Thanks, perfect. So to model what lactate is doing, we began to explore how can we model lactate without actually exercising these animals? How can we pretend that these animals are actually exercising in mice and see what lactate is doing? So what we did is we injected mice with lactate, just like you would be exercising, the same amount that they would have. They were exercising hard and running. And we did this for 10 days, and we had mice that injected with just saline. And then we looked at gene expression changes, and we looked at cell structure changes, especially in astrocytes, and particularly in striatum, which we know in mice is the equivalent of the caudate putamen, which is where we have a loss of dopamine in Parkinson's disease. And what we've shown is, this is very new data, that when we actually just administer lactate itself, we see changes that recapitulate exercise effects alone, just with lactate itself, is able to do the same exact thing, at least at the gene expression levels in astrocytes, that exercise itself is doing. So this to us is showing that lactate as a metabolite, as a form, as a byproduct of metabolism, both peripherally through your muscles and centrally in your brain, is actually able to modify the system and improve metabolism. And we actually see a number of different things when we think of, for example, BDNF, which is a trophic factor that's incredibly important. It's a byproduct of exercise and support for neuroplasticity. Lactate itself increases BDNF expression in the brain of these animals. And we show that when we do this, when we inject this lactate and then we walk these animals so they have activity with the motor circuits, what we see is that only in the areas that exercise is actually working, where we see motor behavior is important. So in the striatum, prefrontal cortex, we see these changes, but in other areas of the brain, we don't see it. So this lactate is being used in a very specific way, in very specific parts of the brain that are important for motor behavior. And so this to us, while we don't think that you should go take some injections of lactate that will fix your problem, or you shouldn't have to exercise, what this shows us is that this one molecule may be one way in which the brain is using the byproduct of exercise as a way to facilitate all of the behavioral changes that we see. And astrocytes themselves, this is a little small here, you can see. So before I showed that exercise is able to change the shape of these cells, which to us says that they are perhaps functionally changing as well. And lactate itself is able to do this as well. Don't worry about what these graphs become complex, but basically it's making these astrocytes more complex, just like what happens with exercise. And this is just with lactate alone. And we can see here these astrocytes are really fundamentally changing. This is a 3D reconstruction of one of these cells, is that it really is expanding its scope, and here it's reaching out to touch a blood vessel. And it's getting more and more complex. And so, again, like I said, lactate itself may be one molecule that we can target that may be important for mediating a lot of these effects, and as an example of how metabolism may be important for all these changes. So this, I don't think you showed this slide, Mike.
Nope.
So this is, again, from another review paper of ours, but looking at what experience-dependent exercise-induced neuroplasticity is doing, you know, there's clearly changes in synapses, like I showed, there's the increase in the structural changes and more connections being made in the brain. We also see changes in brain health, so with trophic factors and blood flow, we talked about that. We do know there's new neurons being born, but metabolism is something that we are beginning to study more and more, and lactate itself may be one way in which the metabolism is changing to allow all of these things to come together to improve circuitry and then improve motor behavior and cognition in Parkinson's disease. That's it. Thank you for your time.